Okay, I have a feeling that we will have waves of people who enter us when they suddenly realize, oh my goodness, it's the time that it is already. Welcome, everyone. We are really excited today to have Noelia Calvo from University of Toronto presenting her work on biological sex, bilingualism, and dementia resilience. I think, Noelia, you can share your screen when you're ready. I think you should be able to do that.
Yes, I'm gonna try now. Can you all see my screen?
Perfect.
Am I in presentation mode?
You're good. You're
The
perfect.
full,
Yeah, you're
okay,
perfect.
great. Thank you so much. And just got to minimize some windows here. Okay, so good morning, everyone. I would like to begin by thanking Professor Judith Kroll for hosting the Bilingualism Mind and Brain Talk series. For the opportunity to present today. I am really excited to share this work with a group of researchers that are so deeply engaged in research on bilingualism. Today, I will be presenting a study examining how biological sex and bilingualism interact in shaping risk and resilience for dementia, particularly among individuals with mild cognitive impairment. This work, in general, aims to contribute to the ongoing discussions about about how bilingual experience influences brain health in aging, and whether those effects may differ between males and females. This project is a result of a collaborative effort that grew out of my two postdoctoral positions, one in bilingualism with Dr. Ellen Bialystok, and another one in women's health with Dr. Gillian Einstein. I was very lucky that I got to collaborate with Dr. Ellen Bialystok, my first mentor here in Canada, pioneer in bilingualism and dementia research, but also Dr. Natalie Phillips, recognized expert in neuropsychology, dementia, and bilingualism research, and also Dr. Gillian Einstein, who is an expert on women's health research. So I'm very grateful to Dr. Bialystok, Dr. Phillips, and Dr. Einstein for their guidance and expertise. So the work brings together insights from all the different labs, and the study that I present today focuses on a research question I developed within that collaboration. So to start setting the stage for this work, I want to briefly review why understanding dementia risk and resilience has become such an important research question. So let's start by looking at some current statistics. Today, more than 46 million people worldwide are living with dementia, and projections suggest that this number could increase to around 130 million by 2050 as populations continue to age. We also know that women are disproportionately affected by Alzheimer's disease compared to men. For example, in Canada, approximately 600,000 individuals are currently living with dementia, and about 72% of those diagnosed are women. However, the sources of these sex differences are not fully understood. A range of biological, social, and cognitive factors have been proposed, but the mechanisms underlying these differences remain an active area of research. So given these large sex differences in dementia, an important next step is to look more specifically at Alzheimer's disease, which is the most common cause of dementia. So Alzheimer's disease is the leading cause of dementia, and it accounts for the majority of cases worldwide. When we look specifically at Alzheimer's disease, we see several notable sex differences. For example, some studies show that or suggest that after diagnosis, women may also experience faster progression of cognitive decline. They also have a higher prevalence for Alzheimer's disease. And for a long time, these differences were often attributed simply to the fact that women tend to live longer than men. However, more recent research suggests that longevity alone does not fully explain the disparity. So because of these, researchers have increasingly emphasized the need to examine both risk and factors and potential also protective or resilience factors that might influence how Alzheimer's disease develops and progresses. So beyond the differences in prevalence and progression, researchers have also identified biological and cognitive differences between males and females in the context of AD. So several studies, for example, suggest that women may also show different underlying patterns of brain pathology associated with AD. For example, women have been found to show greater tau burden or greater accumulation of tau, which is one of the key biomarkers associated with Alzheimer's pathology. Women may also experience faster hippocampal atrophy, which is particularly important because the hippocampus plays a central role in memory formation. And then hormonal factors have also been proposed as a contributing mechanism. In particular, the decline in estrogen following menopause has been linked to changes in brain function and may influence vulnerability to Alzheimer's disease. However, an important and somewhat paradoxical finding is that women often perform better than men on measures of verbal episodic memory, even in the presence of similar pathology or similar levels of pathology. This advantage is particularly relevant because many cognitive screening tools rely heavily on verbal memory. And this may mean that the early cognitive decline in women may sometimes be underestimated or even detected later. Also, this verbal memory advantage has been suggested as a form of cognitive reserve for women. Now, if we look at the biology, one biological factor that may explain some of these differences is 17-beta-estradiol, which is one of the primary forms of estrogen. And exploring how estradiol influences brain function is important for understanding both memory changes across the lifespan and vulnerability to neurodegenerative diseases. Estradiol plays a key role in supporting brain plasticity. So research shows that it can increase the number of neurons, synapses, and overall neural activity, which are all important for efficient communication between neurons. Now, although estradiol is primarily produced in the gonads, it is also synthesized within the brain itself through the conversion of testosterone via the enzyme aromatase. And within the brain, estradiol is produced in several regions that are critical for memory, emotion, and cognitive regulation. So these include areas such as the areas within the limbic system, such as the hippocampus, which supports memory consolidation, the amygdala, which is involved in emotional processing, and the thalamus, which contributes to sensory integration. Now, estradiol is also synthesized in the prefrontal cortex, which plays an important role in executive functions, such as attention, inhibitory control, and decision-making. And importantly, many of these estradiol-sensitive brain regions are also highly vulnerable to neurodegeneration in Alzheimer's disease. So this suggests that estradiol may have neuroprotective effects, potentially helping to preserve neural structure and function through mechanisms such as interactions with BDNF, for example, which supports synaptic growth and memory processes. And this is particularly relevant for the present work because two of these regions, the prefrontal cortex and the hippocampus, are also strongly implicated in bilingual language control and memory processes. Now, let's analyze the case of bilingualism. A growing body of research suggests that bilingualism may act as a form of resilience, as a form of cognitive reserve or a resilience factor by itself. While the precise mechanisms still remain debated, what we do know is that managing two languages across the lifetime consistently engages processes such as inhibitory control, or proactive control, attention, and language control. These processes rely heavily on brain regions like the prefrontal cortex, which plays a central role in supporting executive functioning. Now, because of this sustained cognitive engagement, bilingualism has been proposed to contribute to greater cognitive reserve. And in line with this idea, several studies have found that bilingual individuals may show a later onset of cognitive decline or dementia symptoms compared to monolinguals. And there is also evidence suggesting that bilingualism is associated with structural brain differences, including changes in gray matter in regions like the regions involved in language processing, attention, decision-making, and episodic memory retrieval. However, despite this growing literature, one important question remains largely unexplored. And it is the relationship between bilingualism and biological sex, specifically for resilience for dementia. So the question is, does biological sex influence the relationship between bilingualism and cognitive resilience? And that is the main question for this paper that we published a couple of months ago. So we noticed that gap in the literature in understanding how bilingualism interacts with biological sex to influence AD. And to approach this gap, we asked 3 different research questions. The first one is there interaction between bilingualism and resilience in females and males? If so, does bilingualism offer greater resilience benefits to females or males? And if bilingualism increases resilience more for one sex, what might explain this difference? To address these questions about sex differences in bilingualism-related resilience, we analyzed data from a very well-characterized cohort here in Canada, the Comprehensive Assessment of Neurodegeneration and Dementia, or COMPASS-ND, of the Canadian Consortium of Neurodegeneration and Aging, the CCNA. The study focuses on individuals at the mild cognitive impairment stage, which is critical for examining resilience before full dementia onset. And our cohort included 335 individuals with mild cognitive impairment. The participants include both monolingual and bilingual participants, as well as males and females. And this allows us to examine the potential interactions between sex and language experience. Participants also had biomarker data available. In addition to cognitive assessment, the COMPASS-ND dataset also includes very detailed demographic and clinical information. And this also allows us to control for important factors such as age, education, and general health. Here we have a summary of key demographics and clinical characteristics of our cohort, divided by sex and language group. So the table shows that the average age across groups was very similar, ranging from about 72 to 74 years old. In terms of immigration, a higher proportion of bilingual participants were immigrants compared to monolinguals. And this is not surprising, as bilingualism is often associated with immigrant populations. But it also reflects the broader linguistic landscape in Canada, where bilingualism is relatively common. Our bilingualism score, highlighted here in red, shows a clear separation between bilingual and monolingual groups, as expected. And now if we look at the hormones levels, we see that estradiol concentrations were higher in males than females. This at first was a little bit surprising, but then we also saw that this actually aligns with known biological patterns. So men continue to convert testosterone to estradiol later in life, and they end up having relatively higher levels of both hormones in older age. So they may end up having the benefits, biological benefits from both hormones, estradiol and testosterone. Importantly, there were no significant group differences in global cognitive screening measures, such as the MoCA, the Clinical Dementia Rating, or the Lawton Instrumental Activity of Daily Living scale. However, we did observe differences in logical memory, and in this case, it was the bilingual participants who outperformed monolinguals on this clinical measure, which is a measure of verbal episodic memory. So overall, this table here just shows that the groups were well matched on key demographic and clinical variables, while also showing expected differences in second language proficiency, education, immigration status, sex, esteroids, and logical memory in the case of bilinguals. Now, bilingualism was operationalized in two ways. First, as a categorical variable, distinguishing monolinguals and bilinguals. And then as a continuous bilingualism score reflecting language proficiency across reading, writing, speaking, and listening. Now, although some participants reported additional language, proficiency beyond the second language was generally low, very low. So the analysis just focused on the second language proficiency. In general, we saw that in this cohort, we could consider these bilinguals with some knowledge of a third, fourth, or fifth language, but we just focused on second language proficiency for the analysis. Mild cognitive impairment classification was based on cognitive, functional, and mood assessments within the COMPASS and LEAP protocol. So participants completed several assessments. Cognitive screening was conducted using the MOCA. Global clinical status was measured with the clinical dementia rating. And then neuropsychological testing included the Seurat battery and the Wechsler logical memory delay recall task. We also assessed mood using the geriatric depression scale and functional status using the Lawton-Brody scale. So in general, the neuropsychological assessment captured multiple cognitive domains, including memory, executive function, attention, processing speed, and visual spatial abilities. Biological measures included sex esteroids and dementia biomarkers, circulating levels of E2 and testosterone, estradiol and testosterone, were measured from serum or plasma using standard clinical assays. Blood biomarkers were measured using the single molecule array or SNOR. And this included beta-amyloid 40, 42. We also examined the ratio, beta-amyloid 42-40 ratio. Glial fibrillary acidic protein or GFAP. This was for astrocyte activation. So this is a measure of glial response to brain injury. And also we included NFL for axonal damage. Now, as regards to the analytical approach, comprised of two different phases. First, we conducted group comparisons across sex and bilingualism using ANOVAs and the chi-square tests. And this analysis tested the main effects of sex, the main effect of bilingualism, and also the interaction between sex and bilingualism. And the post-doc comparisons were done using two key tests. Now, the second step was the structural equation model. We use the structural equation modeling to examine relationships among biological sex, bilingual proficiency, circulating levels of sex esteroids, specifically estradiol and testosterone, and demographic factors. So general cognition here was modeled as a latent variable, and this was derived from nonverbal cognitive tests. We use specifically nonverbal cognitive tests with the goal of minimizing language-related bias, particularly in bilingual populations. And verbal memory here, as indexed by the Raybould, was included as a mediator to test whether language-related memory processes might help explain associations between bilingualism, sex-related biology, and cognition. The path coefficients from this model were then used to derive an individual resilience index. And it was more the logic behind this resilience index. So as I said before, we used these path coefficients from the model, and then we computed an individual resilience index by combining bilingual proficiency, sex asteroids, age, education, and immigration. So in general, this index estimates expected cognitive performance based on these biological and demographic factors. So resilience is then defined as the degree to which an individual's observed cognition deviates from that expected level, with higher scores indicating better than expected cognition. The participants were compared based on the resilience scores using bootstrap resampling with 1,000 replicates. And with this approach, what we wanted to do is to shift the focus from whether bilingualism delays dementia to how cognitive resilience might operate across different biological contexts. And these are the results specifically for the first research question. So the first research question is there interaction between bilingualism and resilience in females and males? For this analysis, we only did univariate tests and of the ANOVAs and chi-square tests. So for the cognitive performances, you can see here in these plots, we only observe main effects of sex. Females outperform males on several measures, including verbal memory, verbal episodic memory specifically, and associative memory. Males also had slightly lower scores on executive function as measured by the DPAFS. This was the color word interference task. And now there was no main effect of bilingualism for the cognitive performance, for the cognitive markers. But for the brain markers, bilingualism was associated with smaller gray matter volume in the left and the right temporal lobe. And sex differences in the brain markers were also seen across total and temporal lobe gray matter. In this case, females showing lower volumes than males. And females also had higher GFAP levels, which indicates more neuropathology. In these first models, we did not observe any interaction between sex and bilingualism using this univariate approach. So this left open the question of whether bilingualism might influence cognitive resilience differently in males and females, which we then explored using multivariate approaches. And these are the results to answer, that answer our second question, who benefits more from bilingualism in terms of resilience? So these are the results of the resilience score that was calculated using the path coefficients from the SEM model. So yeah, because no interactions were observed in the univariate analysis, we turned to this more multivariate approach to capture how cognition might differ relative to biological and demographic risk factors. The key findings in general were that bilingualism contributed positively to resilience. Bilingual males show the highest resilience score, and therefore the lowest predicted risk for Alzheimer's disease. Bilingual females have an intermediate phenotype or an intermediate resilience. So it was higher than monolinguals, but lower than bilingual males. And I think this highlights that the cognitive resilience might operate differently across biological contexts. So these are the results of the third question. What mechanisms explain sex differences or might explain sex differences in bilingualism-related resilience? So to investigate these questions, we developed a second structural equation modeling analysis And in this case, we introduced the Ravel or verbal memory as measured by the Ravel as a mediator. So this model examined both direct and indirect effects on general cognition and demonstrated a very good fit to the data. So this indicates that the model accurately represents a relationship among the variables. Specifically, for example, the comparative fit index was 0.97. The Tucker-Lewis index was 0.95, both exceeding the standard cutoff of 0.90 for a good fit. The root mean square error of approximation was 0.04, and the standardized root mean square residual was 0.03, showing low error and close model data correspondence. In general, females show a strong positive effect on verbal memory. Bilingualism also positively influenced verbal memory and had a marginally significant direct effect on cognition. Crucially, the indirect effects of both female sex and bilingualism on cognition through verbal memory were significant, indicating that verbal memory partially mediates this relationship. So conceptually, what the model suggests is that females and bilinguals tend to perform better on verbal memory, specifically on verbal episodic memory. And better episodic memory might be associated with better overall cognitive performance. And part of this association between sex, bilingualism, and cognition is transmitting through this verbal memory, verbal episodic memory pathway. So verbal memory is one core domain that contributes to overall cognitive functioning. And if a group shows an advantage in verbal memory, that advantage might partly account for the higher global cognition scores. We also saw that visuospatial memory was possibly associated with both verbal memory and bilingualism. So this suggests that there might be some interconnected cognitive pathways. And different research has shown that testosterone may influence visuospatial memory in males through its conversion to estradiol by aromatase. So one interpretation could be that this could help explain why bilingual males may rely more on verbal memory, verbal episodic memory, to compensate for the visuospatial decline, contributing to greater cognitive resilience compared to females overall. Now, I think it's important also to highlight here from these models that mediation reflects a statistical pathway consistent with a mechanism, but it does not definitely prove causation. So it's one interpretation. Then we conducted a sensitivity analysis. Here we included, this sensitivity analysis included 505 participants. So these are the 335 participants with mild cognitive impairment from the COMPASS and DEED. We also then included 170 participants with Alzheimer's disease from the COMPASS and DEED cohort. So we included 80 patients because this allows us to test whether the resilience index relates not only to cognitive status within mild cognitive impairment, but also to possibly a progression to clinical dementia and pathology. So in these logistic regression models, the resilience index was the outcome variable, while AD status was the categorical predictor. This setup tested whether resilience scores differ between groups and how strongly resilience is associated with dementia risk. In general, we found that higher resilience scores were strongly linked to lower odds of having Alzheimer's disease. Each unit increase in resilience reduced the odds roughly by over 94%. The linear regressions also showed that resilience predicted better cognitive performance on the MoCA, the SERAT, and the logical memory. And the pathology analysis revealed that higher resilience was associated with lower levels of p-tau 181, GFAP, and NFL. So this indicates a protective effect against neurodegeneration and glial activation. We did not see any association with amyloid biomarkers. So this is the first study to examine resilience by sex in bilingual orals with mild cognitive impairment. So we found that bilingual males show higher resilience scores, which corresponded to lower odds of developing AD. Sex esteroids level appear to influence verbal memory, and verbal memory may play a key role in mediating cognitive decline. For females, a natural verbal memory advantage might serve as a form of cognitive reserve. And in contrast, for males, the interaction between aromatase activity and bilingualism seems to strengthen resilience through verbal memory pathways. So in summary, our findings suggest that sex and bilingualism interact to shape distinct cognitive resilience profiles. This interaction was only revealed through the multivariate approaches, though, not through the group comparisons. And I think that this highlights that cognitive resilience is a very complex biological phenomenon that might not be fully captured with univariate tests or group comparisons. Now, moving from practical implications to broader theoretical insights, these findings might advance our understanding of cognitive reserve as a multidimensional construct. So multiple mechanisms might contribute to cognitive reserve, including verbal ability, bilingualism, and sex esteroids, rather than just a single uniform factor. The effects of these protective factors are context dependent or seem to be context dependent. For example, bilingualism or sex esteroids may benefit some groups more than others, showing that cognitive reserve operates differently across biological and demographic context. We also see language and sex-specific resilience pathways. Bilinguals and monolinguals rely on different cognitive groups, and males and females appear to use distinct cognitive and biological mechanisms. And finally, these results caution against relying just on verbal measures to assess dementia risk, because we saw that females outperform males on verbal episodic memory. And we also saw that bilinguals outperform monolinguals in verbal episodic memory, even when overall resilience differed. So from a clinical perspective, both females and bilinguals demonstrate cognitive reserve, but likely through these different mechanisms. Verbal memory in females and executive function or nonverbal pathways in bilinguals. This might suggest that we might need more personalized strategies depending on the type of reserve an individual relies on. And then importantly, bilingualism and sex esteroid levels could also be considered in clinical assessments and interventions, as higher resilience scores predict better cognitive outcomes and lower AD pathology. Now, from a public health standpoint, promoting bilingualism and targeting modifiable factors such as education, cognitive engagement may help mitigate dementia risk at the population level. But of course, this study has limitations. For example, our SEM models included variables that are relevant here to this Canadian cohort, but other variables may be important in other populations. For example, we did not see a negative effect of immigration for bilingualism, but that might be not the case in other populations, like for example, in the United States. So other models may include other variables that are relevant for their populations. Also, the effects of bilingualism may differ across MCI subtype, for example, amnestic versus multi-domain mild cognitive impairment. In this sense, there is one paper by Ellen, 2015, I think, that actually shows that bilingualism might be more relevant, or the effect, the positive effect or benefit of bilingualism might be more relevant for the amnestic MCI type. And third, well, the cross-sectional design limits our ability to draw causal conclusions. So future studies should investigate how sex, bilingualism, and other cognitive reserve factors interact over time to influence resilience and maybe use longitudinal approaches as well. So in general, our findings suggest that different resilience profiles may influence the progression from LCI to OAD. Bilingualism supports cognitive reserve, particularly in executive function and nonverbal tasks, which we already know. But also in mild cognitive impairment, the verbal memory pathway, specifically the verbal episodic memory pathway, might be important. In males, aromatization of testosterone to estradiol may make them rely more on verbal memory pathways. But again, this is just one interpretation. Females may benefit from a verbal memory advantage, and this might provide a cognitive reserve or additional cognitive reserve. And overall, bilingual males appear to show the greatest resilience compared to females or monolingual males. So I think these results highlight the importance of considering both sex and language background when interpreting cognitive testing, making clinical diagnosis, and designing research studies focused on cognitive resilience. And that is all I have to share with you today. Thank you so much. I will open now the questions. Thanks.
Thank you so very much, Riley. This was a wonderful talk. Do you want to unshare your